Hi, welcome to Biology classes with Lakshmi Chandra. In today's session, let's focus on the next phase of transcription, that is elongation. Having completed initiation in the previous session, let's see what this elongation in transcription means. This is the actual process in which new RNA molecules are getting added up. So this new RNA molecules are getting added up according to the DNA template. This addition of RNA molecules according to the DNA template is not a simple task. This is also aided by a lot of transcription factors. They aid in this process. So this whole process of transcription elongation can be easily studied if you understand it under three heads. Which are those? First one, promoter clearance, followed by checkpoint clearance, and then polymerization. Once you keep the whole process of elongation under these three heads and learn related to one another, it will be easy for you to recapitulate for the examination and write down things very easily and also score good marks. So let's learn those heads one after the other. Come on. Here, first one, see, promoter clearance. Promoter clearance means the RNA polymerase which is there held up in the promoter is now trying to move away from the promoter. Because if it is still kept in the promoter, new RNA molecules cannot be formed according to the template. So in order to form that, the RNA polymerase has to move away from the from the promoter. So that movement of RNA polymerase away from the promoter is called as promoter clearance. And the key player who is doing this promoter clearance is a transcription factor called as the transcription factor H. TF2H, transcription factor 2H. This is the most important and the key player in this promoter clearance. Why so? It has got two very important enzymatic activities. One, it acts as a helicase. Another one, it acts as a kinase. So two activities done by this particular transcription factor. So this is a complex transcription factor. Very important one as well. Now helicase activity, from the word itself, I think you might have understood that it is unwinding the helix, as we have learned in DNA replication. Kinase activity, what is that? Enzyme kinase is the one which is adding phosphate to the substrate. That means it is doing phosphorylation. That means TF2H is also able to add phosphate. Where it is adding the phosphate? Of course, it will add to RNA polymerase II. Only then it can take the RNA polymerase away from the promoter. But to which region of RNA polymerase II this TF2H is adding? Phosphate. That is very important. You have to know that. Phosphate is added in the fifth serine residue. Fifth serine residue. Serine is an amino acid. There will be lots of amino acids in the RNA polymerase. In which part of RNA polymerase? In the tail region of RNA polymerase. So the tail region has got a name that I had taught you in the last session, in the initiation session. Anybody remembers? What is the tail region of RNA polymerase called as? Exactly. It is called as carboxy terminal domain, or in short, it is called as CTD. Carboxy terminal domain. This looks like a tail of RNA polymerase when you draw the structure. So in that tail there are so many amino acids held one after the other. One amino acid among them is serine. And this serine, which is there in the fifth position of that tail, onto that phosphate is getting added. Who is doing that? TF2H is doing that. It is having the kinase activity. So because of that, phosphate is getting added in the fifth serine residue of CTD, that is fifth serine residue of carboxy terminal domain of RNA polymerase II. When that serine residue is getting phosphorylated, RNA polymerase undergoes a drastic change. The transcription factors, the initiation factors which were they held up, they all get withered off, they all get removed from their places. Now RNA polymerase is a bit free of those transcription factors, those initiation factors, it is free. But one transcription factor, that is TF2F, still clinges on to RNA polymerase II. So that again you have to keep it in mind. Which is that transcription factor which still holds on to RNA polymerase? It is TF2F. Okay, so RNA polymerase is free of other transcription factors except TF2F, and so it is free now, so it will slowly move away from the promoter. There is nobody to catch hold of it. That means it is moving away from the promoter, means the promoter region is cleared. So that is promoter clearance. Promoter clearance. Isn't it very simple? The key player is TF2H. That's over. Now comes the next one. After it moves from the promoter toward, as it goes forward in the template in the 3' to 5' direction, new RNA molecule has to be synthesized. That's not so easy. On the way, it will meet some points where this RNA polymerase will be held up for some time. It will stop there for some time, or it will pause there for some time. And it will take its own time, maybe few seconds or few minutes or maybe for hours it will be held up there. But that should not happen. Without any interruption, it should keep on moving forward, and as it is moving forward, new small RNA molecule has to be formed. This is what the cell needs. So what has to be avoided? Those pauses has to be avoided. So those pauses, you call them as the checkpoints. So next phase is checkpoint clearance. The checkpoint has to be surpassed very quickly by this RNA polymerase. And there are many key players which are helping this RNA polymerase to surpass these checkpoints. I'm just introducing to you here those key players, okay? And after introducing to you, surely I'll explain to you regarding each one's functions by specific diagrams. Let me first introduce to you who are the people or who are the members or the factors which is aiding in clearing the checkpoints. First one, ELL. ELL, it is a protein, and this protein is usually associated with the disease leukemia. Okay, that's just an additional information that I am giving you. ELL, it is usually associated with the disease leukemia. This is present in a gene, okay, and its full form is 1119 lysine rich protein. ELL, 1119 lysine rich protein. It is ELL. Next key player in checkpoint clearance is DSIF. DSIF. Here D stands for DRB. I'll tell you what this is, what the DRB is, okay. DSIF is DRB sensitivity inducing factor. DSIF, DRB sensitivity inducing factor. What is DRB? You know about human leukocyte. Leukocyte means white blood cell. Okay. Human leukocyte antigen is there, and in short it is called as HLA. Human leukocyte antigen is, in short it is called as HLA. In this HLA, there is a D-related beta component. HLA has got many components in that. One such component is D-related component. So this D-related component is termed as DR, D-related. And that can have alpha as well as beta part. If it is having beta, you call it as DRB. If it is having the alpha component we are speaking about, we call it as DRA. Okay, so here DRB is HLA D-related beta component. HLA stands for human leukocyte antigen. Human leukocyte antigen, and here DRB stands for HLA D-related beta component. So this DRB sensitivity inducing factor, DSIF, we have to focus on this one. DSIF is very, very, very important in this checkpoint clearance. Next major component is PTEFB. PTEFB. So here P stands for positive. Positive transcription elongation factor B. P stands for positive. So positive transcription elongation factor B. That means it is aiding in elongation. See the next one, NELF. This is negative regulatory elongation factor. NELF is doing a negative regulation. That means it is not allowing the elongation to happen. We will be seeing how these players are actually involved in this. Okay. So PTEFB is there, positive. NELF is there, negative, against. Last one, TF2S. TF2S, transcription factor 2S. Again, a major key player in checkpoint clearance. So in transcription elongation, the major focus that we have to do is how this checkpoint clearance is happening, or how these 5 factors that I have listed here, how they are working in cognition to each other and doing actually the process of elongation. Okay. Now after learning this, we'll understand the last point, that is polymerization. That means how in the 5' to 3' direction RNA molecule is getting formed, and how they are adding according to the complementary strand, how they are getting added up according to the complementary template strand. Okay. So 3 heads: promoter clearance, checkpoint clearance, polymerization. Promoter clearance is very clear to you, I believe. Now let's focus on how checkpoint clearance is taking place and how these members, 5 members listed over here, are actually acting one after the other. Moving on to checkpoint clearance. So checkpoints, as I've told you, they are the pauses in between this elongation phase. The RNA polymerase, they are just not moving at a constant speed. In between it stops. Again it moves. Again it stops. So there is a pausing. This pausing is not uniform. It can go up to say few seconds to few minutes likewise. But that is not actually welcome, isn't it, in this particular step, in this particular elongation. So these checkpoints, another name for these checkpoints is promoter proximal pausing areas. Promoter proximal pausing areas. Now how does RNA polymerase overcome these pauses? That is coming under this particular topic, checkpoint clearance. Okay. So here, as I have already introduced to you the key players of that one. See, first one is ELL. What it does? It suppresses the transient pausing of RNA polymerase. RNA polymerase, it has this transient pausing tendency. That tendency is getting suppressed. It is suppressed by ELL, this particular protein. But there it doesn't end. Next, very important molecule, DSIF. DRB, stimulating inducing factor. DSIF. What it does, it locks the RNA polymerase very tightly to the DNA template. Otherwise, till this time where it was with the promoter. Suddenly what happened? All the transcription factors which were there in the initiation, they have all left. Now the RNA polymerase is very free. So what it can do? It will have the tendency to move away from the DNA template. If it is moving away from the DNA template, the whole process is gone, isn't it? It has to be strictly there with the DNA template. So there should be somebody to hold it very tightly to that DNA template, and that is done by DSIF. So literally it locks the RNA polymerase tightly to the DNA template so that it is not moving away out of the DNA molecule. Now this DSIF has got an important molecule in it. It's called as HSPT5. This is part of DSIF. Okay, HSPT5 is part of DSIF. What this particular molecule does is it anneals back the upstream DNA. That means, just look at the structure. This red color drawn here is DNA. So let me just make it much more clear that DNA by drawing the positions 5 prime to 3 prime, and this is 3 prime to 5 prime. Okay, I have drawn the strands. That is DNA, the red color that I have drawn here, and this blue color structure that is drawn is RNA polymerase. RNA Polymerase II. And this is the tail part of that, or this is the carboxy terminal domain of RNA polymerase, CTD. Okay, now DSIF, I told you it has got a component called as HSPT5, which is aiding in the annealing of the upstream DNA. Upstream DNA means, suppose imagine this is the promoter region. Now RNA polymerase has moved from the promoter a distance forward, it has moved. So this region which is now unwound here by the helicase activity of TFIIH, it is now unwound. It has to rewind back. That means it has to anneal back, isn't it? So that annealing back of the upstream DNA from where the RNA polymerase has moved, that is done by HSPT5 component of DSIF. Now not only this I told you DSIF is very, very important because it can affect transcription in two ways, either positively or negatively. Okay, how negatively? If NELF, I hope you remember, this NELF, if this NELF is coming and attaching to DSIF, see here it is DSIF, and imagine here I am drawing one NELF. This is NELF. I am just writing here NE. Okay, imagine this NELF has come and it has bound itself to RNA polymerase II. Since it's a negative regulator, what happens is RNA polymerase cannot move forward. So if NELF is binding to DSIF, then there becomes a negative regulation. That means elongation stops there. It cannot move forward. Now, in order to have a positive effect to this one, PTEFB, it comes and attaches itself to DSIF. PTEFB comes and attaches to DSIF. And so what happens is, this PTEFB phosphorylates the serine 2 residue. Imagine here you have the serine amino acid. In the second position there is a serine residue. Okay, the serine residue is getting phosphorylated. When the serine residue is getting phosphorylated here, this NELF, which has attached itself to RNA polymerase, now it cannot remain there. Because why? The PTEFB has phosphorylated the serine residue of CTD. So now NELF cannot stay there. It moves out. Now RNA polymerase is free of NELF. That means it is free of negative regulation. So it can easily now move forward. So which molecule is coming and attaching to DSIF? Accordingly, there will be a negative or a positive regulation. Okay, now it is moving forward, slowly it is moving forward, but then the speed with which it is moving forward is not constant. At some areas, it will again stop for a few minutes and only then it will move forward. So there should be a constant speed with which this RNA polymerase is moving forward. That constant speed is aided by another factor, TF2S. So TF2S, transcription factor 2S, aids in constant movement of RNA polymerase II. It helps it to move forward and forward with a constant speed. Not only that, it also has an important activity, that is proofreading activity. It keeps on reading the bases which are getting added on the way. And if there is any mistake, that means if the basis of RNA which is getting added is not complementary to the template strand, By mistake, if accidentally some other bases are getting added, then it will come to know, which is reading the proof, whether it is properly done or not. So on the way, if it finds that there is some mistake, immediately it switches on its hydrolytic activity. Hydrolytic activity means add water molecule and break the bond. Hydrolytic. Lytic means breaking, hydro something related to water. So add water molecule and break the bond. Okay, so when this TF2S is doing the process of proofreading, it finds that some molecules, RNA molecules are not added properly according to the template, then immediately it switches on its proofreading activity. Proofreading activity is not the function not only the function of TF2S, there are many other molecules involved in proofreading. But here TF2S is involved in elongation as well as in proofreading. Okay, so two processes. Now again, if there are more blocks on the path, SII, another protein is there termed as SII, it will help the RNA polymerase to surpass all the blocks. So it has cleared all the checkpoints with the help of ELL, DSIF. DSIF is also involved in the annealing back of the upstream of DNA with the help of HSPT5, followed by TF2S, which gives a constant speed and also proofreading, and then SII is also there to remove if there are blocks, if any. Now then comes the last part, that is the third head that I have told you, that is polymerization. Now let's understand what that is. Coming to the last part of elongation, that is the actual part, that is polymerization. It's very simple. There are no many factors involved and all here. RNA polymerase is doing its task. The RNA polymerase, it is moving in the 3' to 5' direction, isn't it? It is getting moving in the 3' to 5' direction. So just imagine this is the DNA strand and you have the RNA polymerase here, and this RNA polymerase is moving in from the 3' to the 5' direction. So as it is moving from the 3' to 5' direction, new RNA molecule is getting formed. So that new RNA molecule which is getting formed will be complementary to the template strand. It will be complementary to the template strand and it will be formed in the 5' to 3' direction. These 2 points you have to keep it in mind. Very simple points. So first thing, it is complementary to the template strand. So let me just make you understand by just drawing the bases here. Suppose imagine in the DNA you have the bases adenine, thymine, guanine, cytosine, again cytosine, adenine, adenine, thymine. So these are the bases in DNA. Now mRNA is getting formed by RNA polymerase. So which are the two things? It should be complementary and it should be in the 5' to 3' direction. Of course it will be in the 5' direction beginning. Okay, and the bases which is added here should be complementary to the bases in the DNA strand. So we know the base pairing rule, always adenine pairs with thymine and guanine pairs with cytosine. This is the base pairing rule that we have learned in DNA, but we all know that in RNA there is no thymine, instead you have uracil. So if there is adenine in DNA, the complementary nitrogenous base in RNA should be uracil. So I guess you can just say the bases here. Adenine will pair with uracil. Thymine, of course, with adenine, because adenine is there in RNA. There is no question. Guanine with cytosine, cytosine with guanine, again cytosine with guanine. Adenine, I cannot write here thymine because thymine is not there, instead uracil. Thymine is not there in RNA, okay. Then again here uracil, here adenine, and here I have this 3'. So nitrogenous bases are coming. How are they coming? They are coming in the form of RNTPs. What is this RNTP? Ribonucleoside triphosphate. Each of these nitrogenous bases are coming in the form of ribonucleoside triphosphate. What is that? If it is uracil, it will come in the form of UTP. If it is adenine, it will come in the form of ATP, CTP, GTP, likewise. So they are all coming in the form of their respective nucleoside triphosphate, and this triphosphate, hydrolysis of the triphosphate itself will provide energy for the bonding. Which bonding? Phosphodiester bonding. Phosphodiester bonding or phosphodiester linkage. 3' 5' phosphodiester linkage. Now how is that linkage happening? The fifth phosphate of the incoming nucleotide, whichever nucleotide is coming as a new, suppose adenine is coming here, it is coming in the form of ATP, isn't it? It is having triphosphate. So in the fifth position, it is having phosphate. That phosphate, the fifth phosphate of the incoming nucleotide, gets attached to the 3' OH of the existing molecule. So if I just write between these two, uracil is having, it is ending with 3' OH because it has begun with 5'. Okay, it is beginning with 5', ending with 3' OH. Now who is the next one coming? Adenine is coming. Which part of adenine will come? Phosphate of adenine will come. Phosphate of adenine will come and it will form a bonding here. What is the name of the bond? 3', 5' phosphodiester bond. 3' and this is the fifth phosphate and this is phosphodiester bond. Likewise, again, so this adenine will have a 3' OH here free. Again, which is the new one coming? Cytosine is coming. Cytosine's fifth carbon is having a phosphate that will form a bond here. So always there will be a 5' phosphate of the incoming nucleotide is getting bonded to the 3' OH of the existing one. Again 5' phosphate of the incoming, 3' OH of the existing one. So likewise phosphodiester bond is getting formed. And who is coming? Who is governing that? It is governed by the complementarity rule. Okay, so polymerization is very simple. As the RNA polymerase is moving from 3' to 5' direction, based on the complementarity, new and new bases are getting added by phosphodiester linkages. 5' phosphate of the incoming molecule will get attached to the 3' OH of the existing one. So at the end you will get the 3' OH. At the beginning you will get 5'. So polymerization is always in the 5' to 3' direction. And dear viewers, in order to understand clearly regarding what is this 5' phosphate, 3' OH and all, and to have a base of this, we have already dealt with these sessions in the previous sessions, in the previous videos we have already dealt with this. In order to go through that and get a systematic approach of what is DNA structure and all, and to understand these terms, please subscribe the channel, go to the playlist, see one by one the videos, the headings, whichever you feel like watching, watch and support. Thank you all.